It's great to be here and see the success that the DREAM project has become. I've been involved in this since it was literally just a dream of Gustavo and Andrea. And it's really been a privilege now to be involved as a co-organizer from my new home at Sage Bionetworks. One of the main goals of setting up the Breast Cancer Prognosis Challenge was to develop an extensible system that we hope to reuse in the future to host collaborative challenges designed to answer questions seeking to leverage large-scale genomic data sets to infer molecular predictors of cancer phenotypes. And the first application we chose was prediction of breast cancer survival. This is an application of particular translational relevance. As you may know, there's several commercially available diagnostic assays on the market, such as Oncotype DX and Mammoprint in developing molecular prognostic assays for breast cancer. And the success of these applications has led to many published reports of different gene signatures and different approaches for predicting breast cancer survival. And of course, all of them claim to perform better than the alternatives. And there's even meta-analysis studies that say, for example, that most random gene expression signatures are predictive of breast cancer outcome and indeed perform better than many of the published signatures. Now, the reason for this, at least one reason, is something that Gustavo deemed as the self-assessment trap, which refers to the observation that when scientists are tasked both with developing models and evaluating their own models, they either intentionally or unintentionally tend to cast their method in the best light compared to the alternatives, and it's very unclear what are the strengths and weaknesses of each approach, leading in general in the field to molecular diagnostics having difficulty in reproducibility, expense, and standardization. So one way of getting around the self-assessment trap is using the framework that's really been pioneered by us, by the people in this community here, which is using the system of collaborative challenges that essentially separate out the development of the model from the evaluation, which is then done in a blinded fashion based on predefined objective criteria. So this, as we know, this has really been excitingly picking up steam recently. There's been a number of competitions in genomics, with, of course, our favorite being DREAM, and as well as generic platforms for hosting these types of challenges, one of them being Innocentive, which was a partner in one of the DREAM challenges this year. In the challenge that we set up, we tried to develop a number of novel aspects, four in particular, that I'd like to highlight. The first one is that usually in these types of challenges, the way they work is people are asked to submit essentially a vector of predictions which are scored against a held-out data set. This gets around the problem of objective comparison of methods, but we thought that it would be useful to test out a system that didn't just accept results, but actually accepted models submitted as rerunnable source code. This has a number of advantages in terms of transparency. As Stephen mentioned, that's largely our mission at Sage, to promote openness. So setting up a system like this, we thought would really engage the community. People can see each other's models. People can essentially self-police that the best scoring models are legitimate. People can borrow ideas from each other and build off of the best ideas. People can take innovative models and apply them to a different application that uses predictive modeling. People can essentially mix and match the best components of other people's ideas, and people can combine models to create ensembles. And we saw evidence of all of these things, as Erhan will described throughout the course of this challenge. And in this challenge, we actually went to somewhat of an extreme of openness. In fact, every model that was submitted was immediately available for anybody to download. We hosted, any participants are familiar with the look and feel of this real-time leaderboard. Participants were asked to submit models as rerunnable code. This code would be run on the back end of the system and show up on a real-time leaderboard ranked by the test score, and the models would be immediately available for download using our Synapse software system. So hosting the rerunnable code also allows a lot of flexibility in what we could do in the challenge in terms of reusing these different models and applying them to different data sets. We could slice up the data differently to see if the ranks are still robust. At one point during the challenge, some of the participants suggested that we evaluate the models based on a different endpoint. We're able to rerun all the models and see how that changed the scores and the ranks. It also allows us to be quite flexible in terms of how we set up the structure of the competition, which we roughly divided into 3 phases. So the total data set that we used was from the Metabrick cohort from collaborators Carlos Caldas, Annalisa Borensendale, and Sam Aparicio. This contained a total of 2,000 breast cancer samples with gene expression, copy number, and detailed clinical information. And participants were asked to use these data to develop predictors of survival, which was measured at a median of 10 years, which is significantly longer than had been used in similar studies in the past. So we gave out initially 1,000 of the samples for training to participants, held back 500 samples for evaluation on the real-time leaderboard, and the remaining 480 samples, it turned out, were held back for rerunning all of the models that were submitted and evaluating this in this last part of the data to determine the winners that we are announcing today. And you'll see the approaches by Wei Yi from Dimitris Anastasio's lab and Xin Hua were really, really elegant and sophisticated. The second novel aspect that I want to highlight is phase 3 of the challenge, in which we're generating a brand new data set for model evaluation. So this held out 500 samples is a pretty good test in the sense that the participants never saw the data, but you might argue that some of the models might be overfit to, for example, experimental artifacts in the one large data set, and something that would mimic a little bit better what we would see in a clinical application would be a completely different data set generated on an independent patient cohort from a different institution. And fortunately, we were able to, with funding from the Avon Foundation, generate this data set just for the purpose of evaluating the models that were submitted to this competition. So this is actually somewhat of an announcement. If people are on the email list, you would have received an email from Taya Norman this morning announcing that as of today, we're making the full data set of 2,000 samples available, and people will have 3 weeks to play with the data and submit their final models, which will be evaluated in this new data set that nobody's ever seen in the world. It's brand new, just generated, and this will be the final test, independent test of scoring the winners for what we call phase 3 of the challenge. Now, the third novel aspect of our challenge has to do with recognition, with credit. So as Stephen said, we hope to, in the future, move to a world where when somebody goes for a faculty position, they don't just give their list of publications, but they give their scores in a DREAM competition, and they give their code on the leaderboard, and that's, more valuable because people can really see what they did. But currently, publications are still the dominant currency. So fortunately, we found a way with Kelly Lamarca from Science Translational Medicine, who's sitting right there, to bridge this gap and, in fact, award the winner of this third phase of the competition with a publication in Science Translational Medicine. So this is a really forward-thinking and new way of doing things, in the sense that the process of evaluating models actually serves in place of peer review. So there's not an additional peer review. If you win the competition, that is, in a sense, a more rigorous, I would say, evaluation than traditional peer review, and therefore that is essentially the peer review, and the winners are recognized accordingly. The fourth novel aspect was fortunate donation by a partner of Google. And anybody who's participated in this challenge knows that you were given for free access to a large compute space that they essentially donated to support this competition. There's a bunch of lawyerly talk on this slide, but basically they gave away a lot of computing resources so everybody could build models using a standardized and common framework. So these are the four aspects that we thought we wanted to test and pioneer in this competition: the transparency and reproducibility of submitting rerunnable source code, validation in a newly generated data set. DREAM challenges in the past have generated data for validation, but not at this scale. This is a pretty large data set generated just for the purpose of evaluating the challenge. Recognizing the winner through a new kind of peer review process where the challenge is essentially the peer review, and donation of a compute space to level the playing field where everybody can work in the same environment. So what did we find when we opened up this system for people to participate? Well, the first thing we did in order to figure out what we're doing before we released all of this to the community, we wanted to get our act together, and we did one iteration through a smaller group because we didn't really have things working yet, and these people were more tolerant than the rest. And a group called the Federation, which is essentially a group many people are here today, where we designed to pilot these types of experiments in team science. So we were pretty happy with and encouraged by the participation in this pilot experiment, where in about 2 months we got about over 100 models submitted, which we thought were pretty good. And by having all of the models stored and evaluated on using the same metrics, we could post-process the results and assess the characteristics of different models that influence their performance. And we found some pretty interesting trends that we could use to start weaving a story onto what may constitute the best type of model. So roughly speaking, we started with a baseline simple clinical model using a Cox proportional hazard regression model using only the clinical data, which in fact got a pretty good score. That's definitely one of the findings. Actually quite hard to beat this more simplistic models. The first thing people did was use only the clinical data but apply more sophisticated machine learning algorithms like random forest, elastic net boosting, and they were able to beat the Cox proportional hazard model using only the clinical data. When people then incorporated the very high dimensional molecular data, at first they actually did significantly worse. So this only hurt performance. It turned out that it was very difficult to extract the subtle signals from this very high dimensional data, which may have 40,000 or so features, and a naive out-of-the-box machine learning approach actually hurt performance compared to only the clinical data. As the challenge progressed, people got a little bit more sophisticated and were able to eke out a small improvement with the molecular data. And at the end, it turned out that the most successful approaches essentially combined different aspects that performed well throughout the challenge, using all of the clinical data combined with a machine learning approach. I believe it was boosting regression that worked the best. And not just naively selecting all features, but selecting molecular features based on domain-specific prior knowledge. So by combining the molecular features with prior expertise on which ones are likely to be important, and indeed also aggregate features, such as combining information into a genomic instability index, these combined with the molecular data, in fact, gave the increase in performance. So this is what we did in our small group. When we opened up this challenge to the larger community, we were really blown away by the energy, by the participation behind it. We launched the challenge on July 17th. There's about 150 people who participated in that initial call from 27 different countries. And by the end of the challenge, throughout 3 months, there was about 50 teams that were active. They submitted at least 5 models. I think it was 45 teams or so. But more importantly, those teams that were active were really active. So in particular, the real-time leaderboard concept really incentivized people to continue to try to beat each other's methods. And when somebody beat them, a lot of people would try to submit another model and get those improvements. So this real-time feedback at the end of the 3 months resulted in over 1,700 different models that were designed and submitted to the competition. And you'll see in the next few presentations, how really innovative some of these ideas were. A few anecdotes in closing. We tried a few interesting things as experiments throughout this challenge. One of them was to promote the idea of code sharing. We said, what would happen if we gave away a prize? We offered to give $500 to anybody who actually used somebody else's code, made an improvement, and wound up on the top of the leaderboard. After making that announcement within 24 hours, somebody did this and became the new leader, and we were out $1,000, but that's cheap for innovation. We also saw things that we couldn't expect, that we couldn't anticipate, and the crowd just comes up with ideas that we wouldn't have ever thought of. And one of them was a team that built a game on top of our challenge. And they built their own website where you compete against this little avatar named Barney. And basically, you pick a gene, and then Barney picks a gene, and then each time you pick a gene, it goes into a decision tree and scores how well each one predicts survival. And when you or Barney get statistically significantly better than the other, then you're declared the winner. So some really fun innovations on top of this. We've also fortunately got some nice press attention. In the NCI, actually, the current bulletin nicely highlights the DREAM challenge in general, as well as in other venues. Science Translational Medicine has been a great partner. And going forward, we hope that this is just the start of what we can do with this community. It's been great. I think that this is a great model of research. What Gustavo's done and built over the last 7 years is awesome. And now that we have the computational system to host this challenge, we look forward to planning for next year's challenges, in particular with the discussion that we'll have after the talks that are coming up. And it would be great to hear ideas of what we might do next. We've been thinking about some exciting ideas with TCGA and with cell line challenges, and it would be great to hear how we can best be involved. So I'm going to turn it over to Erhan and then to the winners to tell the really interesting parts, the innovations that came out of this challenge. And in closing for me, I'll thank the whole organization at SAGE, in particular Taya Norman, sitting right here. You guys, whoever's participated, are probably well familiar with her. She did an amazing job at coordinating all of the moving parts, which turned out to be pretty complicated, and we were kind of trying to figure it out as we went along. Eric Wang, who's taken over the assessment in the new clinical data set, of course, Gustavo and Erhan, the whole federation, Sam Aparicio, Carlos, and Annalisa, who provided the Metabrick data set, gave us early access before it was published, and of course Dina and Dan, who supported this effort through the ICBP program, and Kelly Lamarco, who's been our great partner at Science Translational Medicine. Thank you.
Thanks, Adam. It's really exciting. So, do we have a few questions? We have time for one or two.
I guess you tried 1,700 different models. So, how much did they learn the test set? So, was the ranking the same between those two test sets, or was it changing a lot?
Erhan has that exact figure, he's great question, and he'll answer that. The bottom line that he'll show is actually it was surprisingly consistent in terms of that the best performing models in the original round continued to do well. So we thought there would be more overfitting, but he'll show the actual data.
Yeah. Well, one obvious question, what we always did in DREAM was combining the results from different teams, right? How was the ensemble approach in this case?
Yeah, so I think we just planted these questions because Erhan also has that result. Again, the bottom line is actually the ensemble models were quite good. We found that this was a main point that we made in the Federation paper, was that if you kind of naively make an ensemble model that combines everything, that model may not do the best, but it does pretty close to the best. Like, that does pretty well. So that's potentially a robust strategy for inferring models that are likely to be quite good. You know, the ensemble model is within like the 85th percentile of best models or something like that. So but he'll show more.
One more quick question.
Oh, the barriers to, well, certainly something we think about a lot. You know, the reward system, as Stephen said, people are punished for sharing. So, you know, the different models that Stephen described are ones that we're pioneering. I actually think that this system is probably the best that we've seen for encouraging team science, in the sense of harnessing people's competitive and therefore collaborative instincts in terms of trying to really outcompete each other. That's been a major, that's been probably the mechanism that we've found to gain the most traction in terms of breaking out of silos and forming more of a community. But that's a loaded question that we can talk about for
I think
hours.
that's a topic we can talk about in the roundtable discussion. But I think we should move on. So, Iran Bilal is going to talk about